My name is Paul Jackson, and as Leon just said, I am your presenter for today. So today we are looking at International GCSE Biology, getting ready to teach. So the aims of this training is for you to sort of hopefully learn more about the GCSE biology specification if you are getting ready to teach this particular specification. The focus of the training will be planning and teaching the course, understanding how the course is assessed and how to prepare your students. Hopefully we'll be able to give you an understanding of how the content is assessed by looking at assessment objectives and mark schemes. And we'll be looking at how to use the Pearson mark schemes. And finally, we will have a look at the kind of support that is available for you. So as Leon said in the introduction, if you have any questions, please put them into the chat. I've got a window open so that I can monitor the chat, and I'll attempt to answer everything as we go along. So, as I said, my name is Paul Jackson. I'm based in the UK. I was a teacher for 40 years. I've been a senior examiner for UK GCSE biology specifications for many years. So I'm involved in writing GCSE papers and the whole marking process. So I've got a lot of experience of the UK GCSE biology specifications and the examinations, and hopefully I'll be able to sort of pass some of that information across to you for the international GCSE biology. So it might be just useful to get an idea of today's audience. So perhaps if you're able to just put a few comments into chat regarding, well, I assume that you will be new to this course, but let me know if you're sort of currently teaching it, or if you are planning on teaching it, for example, next year, or you've just embarked on teaching it, just to give me an idea of the audience. So, you know, how long have you been teaching? Where are you based? That kind of thing. So if I just give you a minute just to perhaps put a couple of things into chat, and that will allow me to, like I said, get to know today's audience. Okay, well, it doesn't look like anyone's been forthcoming, but that's fine. Like I said, if you do have any questions as we go through, then I will answer them. Okay, so thank you. I might struggle to pronounce your name, but thank you, Dr. Amara, teaching at the International School in Tunis. And interesting to note that you used to teach the Cambridge curriculum. So hopefully your experience with Pearson will be a good one. So thank you for that. Okay. And Shia, Shanghai. All right, brilliant. So yeah, it's so we have obviously people perhaps changing from one provider to another, planning to teach, etc. Okay. Yep. So that's great. Thank you for the information. It's just useful to know the audience, because sometimes people join these events and they've already got some experience, but from what you've just shown me in the chat, it looks as though most of you are joining this course for the purpose it's produced, for the purpose it's used for, which is to getting ready to teach. Okay, so let's move forwards. But like I said, if you do have any questions, then please ask. So in terms of, how long have you been teaching, etc. Well, obviously, from what I've just gathered, you haven't been teaching the specification, or you've perhaps just been teaching it literally a very, very short time. Hopefully, in terms of what you will take away, then we will be looking at, you having a better understanding of this Pearson qualification, and hopefully a better idea of how that you can move forwards in terms of teaching it to your students, whether you're a tutor or whether you're a teacher in a school or whatever. Okay, so welcome, and let's begin the presentation for this particular course. So in terms of Pearson or Pearson Edexcel, world's leading learning company and the UK's largest awarding organisation. So they are very well placed to provide qualifications which are aligned to the British educational system. And Pearson have been doing that for a long, long time. So the international heritage stretches back over 150 years. Obviously, things have changed a great deal over that 150 years, but Pearson have had a lot of involvement. And currently, Pearson partners with schools and universities and employers worldwide, and looking at providing qualifications to a very, very large number of students every year, sort of 3.5 million or more. There are a large number of trusted and recognized partners. So approximately 6,500 schools, colleges, and employers globally. And in terms of scripts being marked, over 10 million exam scripts on behalf of the UK Department for Education every year. And obviously on top of that, all the international scripts as well. And Pearson operate in approximately 70 countries worldwide. And from what I've seen in your comments in the chat, we certainly have a worldwide sort of participation today, which is great. So in terms of the GCSE biology, the International GCSE biology specification, the assessment model is 2 examination papers, which have a range of structured multiple choice and extended response questions. So we'll be looking in more detail at these questions later on. In the papers, or in the specification, there is clear mark allocations and topic coverage for each paper. The specification organizes the content by topic. So, for example, cells and enzymes, genetics, ecology, and that will help with your planning. And then we have 3 assessment objectives, which we just abbreviate to AO1, AO2, and AO3. So AO1 is about the recall of knowledge, AO2 is about the application knowledge and understanding, and AO3 is about analysis and evaluation. And for each paper, each of the 2 papers, there is a specific breakdown of the proportion of these skills which are assessed in each of the 2 papers. So in terms of the specification, I'm assuming that some of you have perhaps had at least a glance at the specification, others perhaps not. But as with other specifications, there is a taxonomy of command words. And again, we'll have a look at these later on in the presentation. But the point about the command words is that the students need to be taught what these particular words mean and how they should answer particular questions which use these command words. We've got mathematical skills which are assessed in these papers. And in terms of the International GCSE Biology Specification, math skills make up about 10% of the total assessment. There are core practicals in the specification, which are a key part of what should be taught. And again, we'll have a look at these later on. Students need to understand particular methods, how to deal with results, variables, and safety. And then the specification, another aim of the specification is to develop skills which involve scientific thinking, data literacy, logical reasoning, and communication. So the idea is that students should be developing skills which are useful beyond the exam room. So the specification, if you haven't already looked at it, you just need to have a look on Pearson website. It's easy to access. The specification has a bright sort of orange cover. It was developed for first teaching in 2017. The first exam was 2019. There have been revisions since then, but it's easy to access from the Pearson website, and obviously it's free. So you can download it. As I'm sure many have done that and I would imagine printed it off. So let's have a look at the actual content of the specification. So there are 5 topic areas. So it says there continue to be. So the previous specification had 5 topic areas. We have the nature and variety of living organisms, which includes the characteristics of living organisms and the variety of living organisms. Topic 2, structures and functions in living organisms. So as you can see, this is a very content-heavy topic. So organization, cell structure, biological molecules, movement in and out of cells, so that would be osmosis, diffusion, active transport, nutrition, respiration, gas exchange, transport, excretion, coordination, and response. So there's a question just come up in chat about, are these the same as the modular? So the answer is yes, the content is the same, but obviously the assessment in modular is different. I won't be talking about the modular today. And then we have another question about being the same in the science single award. So I'll try to address these points as we go through a little bit later, looking at the assessment. But if I haven't answered your question, please submit it again. Okay. But like I said, I won't be talking about the modular today. Obviously, modular is something that the modular approach is something that is offered for the International GCSE science specifications. And that's obviously an option that you can look at following this presentation, and it might be something that perhaps suits the students that you teach. But anyway, let's move on. The third topic, reproduction inheritance, you can see that's a small topic by comparison with the second one. So the second topic includes lots of, if you like, traditional biology. moving on to the fourth topic, ecology and environment. So organisms in the environment, feeding relationships, cycles, so nitrogen cycle, carbon cycle, and human influences on the environment. And then topic 5, uses of biological resources, so food production, selective breeding, genetic modification, and cloning. So some of the content in topic 5, students find quite difficult, particularly the sort of genetic modification. Okay, so we'll have a look in a little bit more detail of the different topics. So topic 1, nature and variety of living organisms. So we're looking here at introducing learners to what it means to be alive, how organisms are classified, and the diversity of life. So the topic is split firstly into part A, characteristics of living organisms, a kind of sort of general biology that we teach about key life processes, nutrition, respiration, etc. I'm sure some of you use a mnemonic. Sort of we used to use Mrs. Gren. So M for sort of movement, R for respiration, S for sort of sensitivity, etc. I'm sure you've got your own kind of mnemonic that you might use for students to help learn these different key life processes. Part B of topic 1, the variety of living organisms. So we're looking at the groups of living organisms and the key features of those organisms. So the fact that plants photosynthesize, they have cellulose cell walls, contain starch, nervous controlling animals, in fact they have glycogen, chitin walls in fungi, hyphae, specialist nutrition, saprotrophic nutrition, and then the protoctists such as amoeba and plasmodium, bacteria without a nucleus, circular DNA, an example being pneumococcus, and then viruses, non-living, so HIV and TMV, so tobacco mosaic virus. And in terms of the proportion of marks that topic 1 covers in the exam overall is about 5%. So as you can see, it's quite a small percentage of the overall exam, which is accounted for by topic 1. And as it says in the second bullet point, it's heavily vocabulary and classification based. So students need to learn lots of vocabulary, sort of definitions. So even though it's only 5% of the total exam marks, it's still a very important chunk of marks. And typically topic 1 content might be assessed through multiple choice questions, giving definitions, and short structured answer questions. So perhaps, a question that asks students to perhaps state the difference between plant cells and animal cells, that kind of thing. Comparing, say, a bacterial cell with a plant cell or animal cell, just questions that focus on students knowing specific information about these different groups of organisms. So topic 2, as we saw in the previous slide, there is a large proportion of content here, looking at living organisms, how they're built, the different systems, etc., etc. So in terms of some of the subtopics, there's focus on structure of cells, differences between cells and how cells are specialized, the different biological molecules, carbohydrates, proteins, etc., structure of these things, how substances are moved into and out of cells by diffusion, osmosis, and active transport, photosynthesis in plants, and then sort of diet and digestion in humans, looking at nutrition, aerobic and anaerobic respiration, and ATP production, gas exchange in plant leaves and in lungs in mammals, how leaves are adapted, how lungs are adapted for gas exchange, transport systems in plants and animals, so the xylem and the phloem in plants, blood system uh, in in humans, and the heart, circulation, etc. Excretion, so waste removal in plants, so removing for the gas exchange that relates back to gas exchange in plants. Humans, the kidney and excretion, and then coordination and response. So how the nervous system and the hormonal systems in humans function, and reflexes and homeostasis. We have a question about the textbook. Should we use the latest textbook for definitions for biology? I would say that it's obviously always best to use the most up-to-date version of a textbook if you are able to, but obviously textbooks are expensive. You can always check to see whether there have been any changes in the more recent versions of a textbook compared with an older one. But in general, it is unlikely that definitions will change for particular terms. I guess the only thing that could be different is perhaps there might be an updated list of definitions which include new ones. But in terms of the definitions, there's nothing wrong with the definitions in older versions of the textbook. I say it would be a very expensive exercise if you were replacing textbooks just to get more updated versions of definitions, etc. So the older versions of the textbooks should be fine, but obviously some of the content may be updated for some of the topics. It's always worth, like I said, having a look and making comparisons and making a note of what's perhaps been updated. But in terms of perhaps replacing them, that will be an expensive exercise if you were doing that very regularly. So, as we've already said, topic 2 covers a large number of concepts and practicals. And it's a topic that underpins much of what the students are taught in later topics in the specification. And as it says, this topic accounts for a significant proportion of marks. And topic 2 considers plants and animals, which links with the international primary curriculum and the international A-level curriculum. So this sits bang in the middle and shows progression from primary through to A-level. Topic 3 explores living organisms, how they reproduce, the inheritance of genetic information, how genetic information is expressed, and variety across generations. So we're looking at sexual and asexual reproduction, structures and functions of reproductive systems in plants and humans, the menstrual cycle and roles of hormones. So there will be a link there, for example, with homeostasis and topic 2, fertilization, embryo development, and the placenta and amniotic fluid. And there is a core practical about the germination of seeds. And then the second subsection about inheritance. We're looking at the structure of DNA, genes, chromosomes and the genome, protein synthesis, key terms. So this perhaps links back to the previous question about definitions. Dominant, recessive, genotype, phenotype, etc. non-hybrid crosses, family pedigrees, sex determination, comparing mitosis and meiosis, haploid and diploid, variation mutations, and then the role of mutagens and evolution by natural selection, and then looking at antibiotic resistance as an example of evolutionary pressure. So as it says at the bottom of the slide, there are lots of abstract and technical concepts in topic 3. So using models and diagrams and perhaps simulations can support the students' understanding. And so, some of the command words used in questions which assess topic 3 may be more demanding command words, which require perhaps more in-depth answers and more thought, not just recall, but perhaps application of knowledge as well. Topic 4 looks at living organisms, how they interact with each other and their surroundings, how human activity impacts on ecosystems. So we're looking at organisms in the environment, ecosystems, environmental sampling, biodiversity, feeding relationships, so food webs, trophic levels, energy transfer, carbon and nitrogen cycles, and typically students find the nitrogen cycle more challenging than the carbon cycle, some more difficult concepts. Human impact on the environment, so production of greenhouse gases, global warming, pollution, deforestation. So in terms of trying to, when you're teaching some of these topics, such as the nitrogen cycle and the greenhouse effect, using animations, flow diagrams, that kind of thing, anything to try to, well, firstly, perhaps make the topics a little more interesting, because sometimes the nitrogen cycle doesn't come across as being particularly interesting, even though it's very, very important. But using different methods to teach and to perhaps, say, use animations to liven up the teaching and to also help with the understanding. I mean, I appreciate that these days there are a large number of resources available to you as teachers and to the students. It's just a matter of finding something that you think works well, that you're able to use and embed into your teaching, and to go with it rather than keep looking for, new and perhaps more updated animations, etc. But videos as well, that kind of thing. There are lots of things available, as I'm sure you're aware. Practical skills, so measuring population size, biodiversity. I know that for sort of international teachers, doing what we might typically refer to as fieldwork in the UK can be more challenging. Perhaps you could just let me know in chat if that's the case. So, we in the UK, we might have school fields and so on that we can easily go outside and do a little bit of ecological sampling to teach students techniques of how to sample vegetation or animals. But like I said, I know that for international teachers, that can be more of a challenge. So perhaps you could just let me know in chat if that's the case. And also, I appreciate that some of the core practicals in the specification may be more difficult for some of you to deliver than others. I know that sometimes there can be difficulty in getting hold of different reagents, for example. I know that when I've spoken to international teachers before, Getting hold of enzymes, for example, can prove challenging. So that then obviously means that it's difficult to teach some of these skills from a practical point of view. Okay, moving on to the fifth topic, using biological resources. So this is looking at food production, so the use of glass houses, the use of fertilizers, pest control, fermenters, fish farming. And again, I appreciate that for international students, some of these methods of food production will be unfamiliar to them, depending on whereabouts in the world you are. Selective breeding, um, genetic modification, so using enzymes, vectors, recombinant DNA to enhance organisms, and then micropropagation and animal cloning. Obviously, Dolly the sheep is sort of very old technology, as it were. I mean, it's a long time since Dolly the sheep was cloned, but it's still used as an example, obviously, in textbooks. So the point of topic 5 is to combine real world applications with some complex concepts, such as genetic engineering. Typically, these are areas of the specification that students often find very difficult. And again, with some of the other areas of the specification, using different methods, visual aids, etc., to try to simplify the content without sort of diminishing the quality of what you're teaching, but any method of simplifying and improving the way that students learn these topics is useful. So we've just looked at a summary of the 5 topics in the specification. So you may not have, as we know, you may not have taught specification as yet, but perhaps you could just put into chat the topics you think the students you teach and have taught would find the most challenging. What have you done to help your students in the past? And can you give any examples of techniques, etc., that you've used? So I'll just give a couple of minutes for you to perhaps put some thoughts into chat, and I'll just go through them as they appear. But it's useful to share ideas. So like I said, I'll give you a couple of minutes perhaps to get a few ideas together and put them into chat so that we can share them. Okay, so Lucia or Lucia, might be mispronouncing your name. So perhaps you could just say why your students might find the ecology and the environment perhaps more challenging, please. Anyone else got anything to share? Okay, so we got challenges with describing structures of cell organelles. I mean, certainly at the international GCSE, so obviously detailed structure of organelles would not be required. That's more for the international A level, but clearly sort of knowledge of the organelles is important in the international GCSE. Any other comments? yeah, so clear diagrams, videos. I think diagrams with annotations, um, sort of a tried and tested method of sharing information and helping students to learn. And obviously, yeah, videos. Um, there are lots and lots of sources of short videos that can be used to help clarify different topics. Any other comments? Well, I think just to summarize this, clearly, when you've got a group of students, you do tend to find out what or which of the topics or subtopics the students do find challenging, and you will, put some emphasis into helping those students to learn those concepts, particularly when it comes to revision and preparation for exams. And there are always topics that require the extra bit of emphasis, particularly during times for revision. Okay, so just a couple more comments just to go through. So yeah, so a comment about the carbon and nitrogen cycles, sort of visualizing, because obviously these are the carbon cycle and the nitrogen cycle are conceptually, some students find it very difficult to be able to follow what's actually going on. So using any kind of diagram or video to help them is good. And then we have a comment about practicals. Students not always following safety procedures when engaging with chemicals. Um, we'll have a look later on at practicals, so we can perhaps come back to this problem of safety procedures, etc., a little bit later on in the presentation. So thank you for your comments. Um, so it's helpful to share um, how we deal with issues of students finding concepts difficult and how we can help them to particularly with the more abstract concepts. Okay, so there was a question earlier about the modular assessment structure. And as I said at the time, the purpose of today is to look at the linear structure. The modular, I think, if I'm correct, the first assessment will be next year, um, possibly November this year. I can't remember off the top of my head, but the modular assessment will be, the first assessments will be in this academic year, as far as I'm aware. I said today we're looking at the linear approach. Okay, so let's have a look. So the international GCSE that we're talking about today, the linear model, we have written untiered examinations. In the UK GCSEs, we have tiered examinations. So we have a foundation tier and we have a higher tier. So the foundation tier has a maximum grade that can be awarded of grade 5. So the foundation tier in the UK specification is grade 1 to grade 5. And then the higher tier would be grade 4 to grade 9. Obviously, so there's some overlap between them. And in fact, in the UK GCSE science qualifications, there is some overlap between the questions in the foundation paper and the higher paper. So there is a small proportion of foundation paper questions and higher paper questions which are the same. They overlap. So coming back to the international GCSE, the students sit the same papers, which are graded from 9 to 1. So they are not tiered, they are untiered. And the questions are written to be accessible across the full ability range. In other words, from grade 1 up to grade 9. So as a centre, you can choose a linear model or a modular model. The content is the same. There's no practical examination. Practical skills are assessed within the written papers. So as we said earlier, and as we'll have a look at a bit later, there are a number of core practicals that the students would be expected to be taught as part of the sort of normal teaching for each topic. And those core practicals can be assessed through the written exam papers. Both of the papers have similar styles of questions, and paper 2 has content that is not assessed in paper 1. So I'll explain that in a moment about the additional content. And the types of question styles that we have are multiple choice, short answer. So a short answer might be a single line. The command word might be something like state or give, and there might be one answer line, because all we're looking for is perhaps a single word answer. Then we have longer answer questions, or what we call extended open response questions. So they may be, a short answer question may be 1 to 3 marks. A long answer question might be 4 marks, up to 6 marks. And some questions are based on using data, whether that's a table of data or perhaps a graph. And we have planning questions, and the planning questions obviously tend to link to the core practicals that we expect the students to have been taught. So in the linear route, the assessments are taken in one exam series, and the grades are calculated on the raw marks for the papers. If students are resitting, they can resit assessments for all units together in one exam series. And the students receive a grade that's calculated at the end of the exam series in which they sat their assessment. So 2025 students will have sat their exams in the academic year 24-25. So that's the exam series. So they'll have done their 2 papers, and their grade is based on the marks they received for the 2 papers in that exam series. So looking in a bit more detail at the 2 papers. So both papers are assessed externally. They're available twice a year, so November and June. So the international GCSE was first assessed in June 2019. And you can see that paper 1 is just over 60% of the total for the international GCSE award. The content, so paper 1 can have questions based on any of the 5 topics, but not content that is bold in the specification, and not content that has a B reference after it. And paper 1 is 2 hours long, marked by Pearson. I'll talk more about marking later on. The paper has a total of 110 marks. We've got the different types of question that we've already mentioned, and students can use a calculator. Paper 2, again, it's available in 2 windows, November and June. It's worth a total of just under 40% of the total IGCSE. Paper 2 can assess all of the content in the specification. So some of the content in the specification is in bold. Some of it has a B reference. So that content can be assessed in paper 2. And again, just as in paper 1, the questions can be based on any of the 5 topics. The statements in bold cover some of the subtopics in greater depth. So when you prepare students for the exams, you know and they know that they will not be assessed on any of the bold content in the specification, nor any of the content that has that B reference next to it. The assessment it's a shorter written exam, 1 hour and 15 minutes. Again, set and marked by Pearson. It's worth 70 marks. And just as in paper 1, there are a mix of different styles of questions, and students can use a calculator. So please ask if you get any questions about the specification and the papers. So in terms of a comparison, both paper 1 and paper 2 externally assessed by Pearson, available in 2 windows, so November and June. Different styles of question. So the extended open response are those questions which are worth between 4 and 6 marks. Calculator can be used. Questions can be from any topic area. So here's a visual summary. The paper 1, you'll notice, has 2 codes. And the second code, 4SD0/1B, that code is for the double award. So paper 1, International GCSE Biology paper 1, is also a common paper with the double award for International GCSE Science. So I think we've probably talked through most of the differences, similarities and differences. The key one perhaps to bear in mind is that paper 2, in terms of depth of coverage, paper 2 will have greater depth of coverage on some subtopics. So here we have a little excerpt from the specification, topic 2, and you will see that at the bottom of this sort of chunk of specification points, so the points with like 2.2, these are what we call specification points. So 2.5 and 2.6, they're in bold. They've got the B suffix. So that means that 2.5 and 2.6 can only be assessed in paper 2. 2.2, 2.3, 2.4, they could be assessed in paper 1 or paper 2. So when you are looking at the specification, anything in bold with the letter B after the specification point number, that's only going to be in paper 2. So that's a way of interpreting the specification and knowing what will be on paper 1, what will be on paper 2. So in terms of the questions and how the papers are put together, so the questions are sequenced to increase in demand. So in other words, we start off with relatively straightforward questions, perhaps using a little bit more or involving more recall. And then as you get towards the end of the paper, they become more demanding and help us to differentiate between the grades. So you wouldn't necessarily expect perhaps a grade 4 student, for example, to be scoring many marks on some of the later questions in the paper because they're focused on or targeted on the higher grades. Also, within an individual question, we have what we call ramping, which in other words, as you go through the question, it might start off easy and then the later parts of that question become more difficult. So you start with perhaps a little bit of recall, give the name of, and then you move on to application of knowledge and possibly evaluation. Evaluation is a skill that many students find very, very difficult. So the general structure of the papers means that we can assess the full range of abilities from grade 1 to grade 9. And the papers are written to be accessible. So even though a student might not find or might find a particular part of a question difficult, it might still be accessible to them. And they may be able to pick up the odd mark, even if they don't perhaps have a full understanding of what the question is actually asking of them. So in terms of preparing students, they need to pace themselves. So the earlier questions, yes, they might feel easier, but there is a demand, and the more challenging questions require more time to be spent on them, trying to decipher perhaps information and to use and apply their knowledge of particular topics to answer some of those questions later on. So we have a question: how is marking done if they only answer part of a long question? So we will give credit for any correct points. Hopefully this particular question will be answered. I keep saying this, and I'll answer it, later on. We've got some specific areas of the presentation later on that look at marking. And as I alluded to in my introduction, I have many, many years of experience of marking, because as a principal examiner, it's part of my role to, sort of be involved in writing papers, producing the mark schemes, and monitoring the marking during an exam season. So in terms of answering only part of a long question, then, like I said, we will give credit for anything which is positive and matches what's on the mark scheme. So if only part of a long question is answered, if there's something creditworthy in that answer, then the students will get credit for it, and we will mark accordingly. So in terms of paper 2, the questions begin with what we might call a comprehension style question to set a scene. So this is always the same with paper 2. The students are given a short passage or some stimulus material, and then the questions that follow are based on that text, but the students are expected to use their knowledge from across the specification. Many students think, oh, the answers are in the passage of information. Well, perhaps some of them are, but the idea is that the students use and apply their knowledge and understanding from across the whole specification. So the idea of the passage of information is to set a scene, provide some stimulus material that can be used as the focus for the question. So these questions are often synoptic. In other words, they link knowledge from different topics, for example, topic 1 and topic 2, or topic 2, 3, and 4, whatever. So the questions might be synoptic. The context of the passage of information, the stimulus material, may be unfamiliar, but all the answers are accessible through what the students have already been taught. So they should be taught to appreciate that the information, the beginning of paper 2, has something that they may never have come across before, but the actual biology content of that passage is just something that they have been taught, but not in that particular context. That's something that always tends to throw some students. They don't appreciate that they've been taught everything they need to know. It's just that the situation is unfamiliar. So the more experience and practice students have had in terms of practicing on some of these past papers, paper 2, looking at scenarios and the different questions linked to that scenario. The more practice, the better. So here we have I'm not going to go through it, but we have an example of a passage of information. So we've got a whole page of information given the title. This one is given the title Extinctions. And we've got line reference numbers. So this is about Megalodon. The students are told it's an animal. And we have a multiple choice question, which is assessing students' knowledge of features of different types of organism. So this links right back to topic 1, knowing about the different groups of organisms. So they're told that yeast is a fungus. So they're looking at comparing an animal with a fungus. We have a slightly more complex multiple choice question. But that's the way in which students they're expected to apply their knowledge. And then we're looking at the part B of the question is explaining something about evolution. So again, all they have to do is use the knowledge of evolution that they've been taught and apply it to this unfamiliar situation. So this is typical of the beginning of a paper 2. Okay, so do we have any questions about, so we've obviously had one question pop up about marking long answer questions, and as I said, we'll have a look more about marking a bit later on. If you do have any questions, please put them into chat. Okay, so the question just come up. Some students use short terms of different things when they're describing structures. So ER for endoplasmic reticulum. Is it acceptable for the exam? Good question. Some abbreviated terms are just used, have standard use. So for example, ATP. We would just accept ATP. We're not expecting students to be writing adenosine triphosphate. ATP is acceptable. ER is Again, general, I mean, ER for endoplasmic reticulum. In context, that is acceptable. With some terms, the advice is that the students should, the first time that the term is used, they should write it in full and put the abbreviation in a bracket afterwards. And from there on afterwards, just to use the abbreviation. So if it's an accepted abbreviation that you often see in a textbook, then that's fine. If it's something the students perhaps made up, then, like I said, they would need to write the term in full and then put their abbreviation in brackets afterwards. And from there on afterwards, use the abbreviation. And then we have another question about difference in marking AO2 and AO3. We'll be looking at mark schemes later on, but if I just say now that the only difference in marking AO2 and AO3 is the structure of the mark scheme. They're points based, so in a sense there is no difference. It's just about the structure of the mark scheme. And as I said, we've got a section of mark schemes a little bit later on. But thank you for these questions. Hopefully I've answered the one about using abbreviations. So like I said, if it's a standard accepted abbreviation, fine. If it's not, then write the word in full, put the abbreviation afterwards. Okay, then we have another question. Using a definition from another provider, such as Cambridge. So obviously there's a wide variety of ways in which a definition can be written. If the definition is correct, it doesn't matter how it's written. If it's written in a simple way or a more complex way, if the definition is correct, it doesn't actually matter how it's written. So the source of the definition doesn't matter. It's if it's correct or not. So if a student's using simple language, but the language is correct, it's using the correct biological terms, then that's fine. If a student decides to use more complex language to give a definition, again, that's fine as long as it's correct. So any definition is acceptable, however complex, as long as the content of the definition is correct. Okay, so thank you for those questions. So I hope they've answered them. If I haven't, please perhaps raise another question. Like I said, I am monitoring them. Okay, so assessment objectives. There are 3 assessment objectives, and obviously one of the questions a few minutes ago alluded to AO2 and AO3. So the assessment objectives, very important, and they're important in the way that we write the questions. So there are 3 assessment objectives, or 3 AOs. In terms of the proportion of marks for each AO, AO1, the knowledge and understanding, between 38 and 42% of the paper, of the qualification overall. AO2 is the same, and AO3 is much smaller, 19 to 21%. So AO1 and AO2 have the same weighting, and AO3 is approximately half of the weighting of AO1 and AO2. So AO1 is about recalling facts, definitions, concepts. AO2 is the application of understanding to unfamiliar contexts, such as the AO2, the paper 2, as we were talking about a little while ago. Interpreting data, that's AO2, and AO3, experimental skills, analysis, evaluation of data and methods. So it involves interpreting information, drawing conclusions, evaluating evidence and methods. So this is AO3 is the focus of the practical content of the specification. So this is not exclusively, but AO3 is where a large proportion of the marks assessing the practical skills would be. So all questions are designed to assess the assessment objectives. And I think there's always there's a tendency perhaps that we over-focus on AO1. And as you can see from this breakdown of marks, AO2 has exactly the same weighting as AO1. So learning facts and definitions, etc., is very important, but that should not overshadow the application of understanding and the analysis, etc., of data, drawing conclusions. So sometimes a command word in a question will give a big hint as to which assessment objective is being assessed. But both papers assess all three assessment objectives. So, as we've already said, AO1 is about demonstrating knowledge and understanding. It's about recalling facts. It's about giving definitions and so on. Students like it because the ones who like to, learn facts, etc., and recall them, if they can do that, they think they've got good knowledge. They tend to be sort of predictable types of question and easy enough to prepare for. But having a good knowledge and understanding of facts, etc., won't ensure that students get top marks, because we're also looking at the application and analysis skills to get the top marks. So having good AO1 skills will get students so far, but it won't get them to the very top grades. We have an example here of a typical AO1 question. So diagram of a cell, we can see some organelles in the cell. It's a human cell, and we've got a simple, which of the label structures is a microvillus? So it's simple recall. Students have to learn the structures of a cell, and here they're identifying where the microvillus is. So typical multiple choice question used to assess the AO1 content. And then we have another one. Describe how a sample of urine could be tested for glucose. So again, recall of facts. Students need to recall how glucose is tested for. So the context is in terms of glucose in urine, but the knowledge that students need to use is the glucose test, using Edexcel solution to test for glucose. So two typical AO1 questions. AO2, the application of knowledge to familiar and unfamiliar contexts. So we have qualitative and quantitative analysis. So qualitative might be, this is bigger than that, and quantitative is using numbers. So typically we're looking at data, we're looking at graphs, we're doing calculations. The command words, I implied this earlier, but command words used for AO2 may be slightly different to those for AO1. So suggest, explain, evaluate. So these words or these command words imply it could be an AO2 question. Sometimes these questions are or come across as being unfamiliar to the students, but it's just about using the knowledge that they've been taught for a particular topic. And sometimes AO2 requires deeper thinking, and they may require different or a number of steps in their response. So typically an explain question might start off with, they might be given some data and the question might be, explain what the data shows about so-and-so. And so students might perhaps identify some sort of trend or pattern in data. They might start off by commenting on that, and then they need to give reasons. So different steps in their answer to be able to access full marks. So sometimes students leave an exam and they have a moan saying that, oh, you didn't teach us about this, you didn't teach us about that. But the whole point is circulatory systems, human circulatory systems, they will have been taught. So it's about applying knowledge of one to another. So as we've already said, we can use models and animations, different methods to improve students' ability to answer AO2 type questions. More practice, the better, so that students learn to make links across topics. The whole point of an AO2 type question is to make students think, to look at a situation and think, okay, what do I know about this? How can I, what knowledge have I got that I can apply to this situation? So it's not just recalling facts, it's applying those facts to a different situation to which they might be familiar. So we have this question here. We've got a diagram of a frog heart. Question tells the students that it's different to a human heart. And then we've got a question linked to it. Humans can run for long periods of time. Frogs can only move for short periods of time. Explain how the structure of the heart of the frog means that it's unable to move for long periods of time. So students are expected to look at that diagram and think, well, okay, how is this different to the human heart? How is this going to mean that a frog cannot move for long periods of time? So they can see that there's only one ventricle. They can see that blood mixes. So the blood from the lungs and the blood from the body is mixing. And when the blood is pumped out of the heart, it's going to the lungs, the body, and the skin. You can see that there's no valve when the blood exits the heart. So there could be backflow. So the fact that the blood is mixing, there's going to be less oxygenation or less oxygenated blood reaching muscles, for example. So the idea of the question is for students to think, to make a comparison of the human heart with this diagram of the frog heart and think, okay, how is that going to mean that the frogs can't move? So they've got to think about oxygenation of the blood, the fact that it mixes, and the fact that richly oxygenated blood isn't going to reach the muscles. So that's probably quite a challenging AO2 question, and you can see that it's worth 5 marks. So 5 points, 5 marks. So not an easy question, but that is an example of an AO2 question, applying knowledge. Just going back to that question. I'll just go back to the previous slide. So we have a question, any scientific explanation, can it be accepted? So an explanation which is relevant about, for example, the structure of the heart, the fact that blood is mixing, the fact that there's no semilunar valves, the fact there'll be some backflow, and if, for example, less oxygenated blood is going to the body or muscles, then there'll be less respiration, that kind of thing. So relevant explanations will be credited. So explanations that link to the structure of the heart, explanations that link to respiration. Obviously, explanations that are not relevant would not be given credit. On the mark scheme, there were probably about 10 different points which could be given credit, and the students need 5 to attain full marks on this particular question. So relevant explanation, credit worthy. Correct but irrelevant explanation would not gain credit. So moving on to AO3, analysis and evaluation. We're looking at data, we're looking at methods, we're looking at experimental design. Students have to make judgments about whether some data is valid, if it's reliable, if it's accurate. The questions can link to practicals, but not exclusively the core practicals. It could be another practical. We're looking at variables, using controls, interpreting data, making conclusions. There may be questions about how a method could be improved, what are the limitations of a method? And some of these questions use the higher level command words. So higher level command words include evaluate, discuss, justify. So when a student is faced with some of these command words, they often are unsure about how they should proceed with their answers. So this is why it's important to teach the definitions of command words, and we'll have a look at that in due course. Okay, so students need to be confident with different aspects of variables, how error can be introduced into making measurements and so on, how we can improve investigations, and how to interpret data, and how to perhaps think about where the anomalies might be in data and how anomalies could be eliminated. So AO3 is not just about doing a practical. Students need to think more like a scientist and be critical. Think about how can we improve things? How could we design a practical better? So a typical AO3 question. Design an investigation to find out if a change in breathing rate also produces a change in heart rate. Include experimental details and write in full sentences. So this is a 6-mark extended open response question. So we'll be looking at different things. Obviously, the students would need to take measurements of breathing rate. They need to take measurements of heart rate, so they need to explain that. There would need to be perhaps some indication that some exercise would take place. How long would the exercise take place for? What kind of exercise? You need to think about, do you need to repeat measurements? what needs to be controlled, etc. So the type of experimental design question doesn't matter. Students need to be taught particular ways of addressing these questions, and we'll have a look at that. Got a question about right. So I'm just looking at a question in chat. The latest question from Initials AP, I will answer that in due course. Then we've got another question about why we place test tubes in a water bath for 10 minutes before mixing content. So this could relate to, for example, an enzyme type question. So it's about the equilibration of temperatures, that kind of thing. So this question involves students perhaps having first-hand experience of a practical and why you do particular things using a water bath. So first-hand experience of practicals is important. Okay, so question types and command words. As we've already said, there are different types of questions, so I won't go back over these. But short answer questions, typically from 1 to 3 marks, use the command words describe, suggest, explain. They need short, concise answers, possibly even one word. And the longer response questions might involve, again, explain, evaluate, discuss, or even plan. So longer answers need more detail, multi-point answers, often linking things together. And experimental planning often uses, we use the acronym CORMS. If you're not familiar with that, I'll go through that in a moment. But planning experiments requires students to think about the actual method, variables, using controls. So questions like this test understanding of practicals, knowledge of practicals. Maths-based questions might involve calculation, drawing a graph, analyzing data. So sometimes a calculation might be assessed under AO2. And then in paper 2, that sort of passage of information requires some use of synoptic understanding, which we've already talked about. So that paper 2 question often assesses across different topics. So students need to be experiencing the different styles of question throughout the course and not perhaps end loading different, or perhaps the more difficult types of question to the end of the course nearer to the exams. It's perhaps better to expose students to all styles of question early and to be able to sort of teach them how to answer these questions as they're going along. Multiple choice are not necessarily just easy. We saw from that example earlier on related to paper 2, they can be quite challenging and they do involve application and interpretation. So the more demanding multiple choice questions could be assessing higher grades, and the more simple recall type AO1 questions might be assessing a lower grade. So in paper 1, there can be up to 10 multiple choice questions, and in paper 2, up to 5. So that's a fair proportion of the marks. And you need to bear in mind that only 15% of the total marks are for pure recall. So some of the multiple choice questions do require deeper thinking. They're not always the easy option. And students need to be good at interpreting data, thinking about the distractors. Often you see students cross out distractors which are clearly incorrect on the question papers. And they need to use some logic. So using multiple choice questions is quite handy for just getting to grips with do the students know these particular facts, and sometimes they're good for identifying where there are misconceptions. So we've got here just some examples. We've seen one already. So this is the one on the left, which label gland produces LH. So the students just need to recall knowledge of where the different hormones are produced. And then the question on the right is looking at sequence of DNA. So this is looking at base pairing. So again, it's just recalling knowledge and I suppose applying knowledge in the right-hand example using about DNA. So typical questions, multiple choice questions, assessing content. Okay. So the short answer questions between 1 and 3 marks. So we need short answers, they need to be concise. So 1 word often is okay. So here we've got 2 examples. Biological consequences of sulfur dioxide release. Students would need to perhaps mention acid rain. It is acceptable to use bullet points. So that helps students to write perhaps more concise answers. The second example here, 3 differences between asexual and sexual reproduction. So again, it's stating these differences. Short statements. You can see that there are 3 lines for each of the 3 differences, but students might only use 1 line for each. So concise answers that hit the spot. So there's 1 mark for each difference between asexual and sexual reproduction. One thing that we often see is that students repeat the stem of the question, which is something that you can sort of discourage as you use practice examples. And then the longer answer questions. So the mark schemes are points based, not level based. That means that basically it's 1 mark per point. Level based mark schemes are used at A level, for example. And we also use them in the UK GCSEs as well for a very, very small number of questions, but not in the international GCSE biology. So again, we're looking for precise, accurate scientific language. Students can use bullet points, as I've already said. It's about clarity. Students need to look at how many marks that are available, not the number of lines. So for example, a 6-mark question might have, I don't know, perhaps 12 lines available. Students don't have to use all those lines. Concise answer covering perhaps 4 lines could achieve full marks. In terms of spelling and terminology, phonetic spelling is fine, unless there could be confusion with another word. So we see many variations of photosynthesis, for example. That's okay. One that we do not allow is some sort of combination of mitosis and meiosis, because the word that you see in front of you, the mitosis, because there's an E after the M, students might have been thinking about meiosis. So that's something that we would not accept. But many phonetic spellings are acceptable. And as you appreciate, we do see a very, very large number of different types of phonetic spelling. So a couple of examples of these longer answer questions. One about carbon dioxide in the glass house. So this will be topic 5, design and investigation. So this is with crop yield. So it's applying knowledge, 6 marks. So there'll be about 12 lines available. And then we've got the second example, explain how vaccination protects the body from disease, 4 marks. So that's more of a recall question, whereas the first one here is experimental design. So 2 very, very different questions. So any 4 relevant points about vaccination will be credited, and the other question, experimental design, which so we'll come on to a little bit later. Okay, now there are a couple of questions in chat which I haven't looked at. I would just like to take a short pause for 5 or 6 minutes just to get a drink to lubricate my throat. And in that time, I will have a look at the questions that are in chat and attempt to answer them. And then we'll continue in just a few minutes. So I will continue at 10:40. So I'm just taking a 5 or 6 minute pause, and I will continue at, by my time, 10:40. So 6 minutes time. So before we continue, 3 questions for me to answer, which are in chat. The first one was, or is, if a student studies the Pearson GCSE biology curriculum, would they be able to take like the ordinary level, O-level exam somewhere else in the world? So I am not 100% familiar with the, if you like, the international O-level. If the content of the 2 specifications is the same, then learning the Pearson IGCSE specification would be okay. But you would have to look at the O-level specification and make a comparison with the international GCSE specification, because there could be differences in what's taught. So if they're taught the international GCSE qualification, there may be things which are in addition to what's in the other specification, or there could be things missing. So you'd have to make a comparison. And so I can't fully answer that here and now. It's something that you could, at the end of the presentation, there is some information about who you can contact and ask questions. So that could be a question that you could ask of the Pearson representative. We have another question about, do we evaluate grammar? The short answer is no, we don't. Many students have poor grammar. As long as they're getting the point across, then that's fine. We don't knock off any marks for poor grammar. And then we have another question about detailed answers for AO3 for connecting concepts. Well, yes, giving detailed answers is fine. Sometimes, though, students sort of tie themselves up in knots writing complicated sentences. I would always recommend, even for a longer answer question, writing relatively short, well-structured sentences is a better way of addressing a complicated concept than trying to write very long, convoluted sentences. Because, like I said, students often tie themselves up in knots. And sometimes they sort of shoot themselves in the foot. So they'll start off by writing a sentence and something is correct. Then further on in the sentence, they'll write something which is incorrect. So the 2 things negate each other. So yes, it's fine, write it in as much detail as you want, But like I said, I would often recommend that students don't and just write in short, well-structured sentences. Okay, so we're still looking at AO3. One thing that you may or may not know about is using what we call QORMs. So this is a very, very effective way of planning an investigation. And I've always found that international students are good at this. They use this framework and they can apply it to unfamiliar situations to plan an investigation. So the QORMS acronym, sometimes we have or people put extra letters in, so there might be perhaps two M's, and you'll see why in a moment. So C is what are you going to change? So whatever the scenario is, what will be changed, what will be kept the same. What is the organism? Or it might not be an organism, it might be perhaps an enzyme. So in this, we've got an example here, fish. So it needs to be something which is specific. It needs to be, use the same species, all which are the same mass. So here, an example of 10 grams. So the organism, it might be an organism, it might be, could be an enzyme or some other sort of chemical. So that's the O. The R is repeating. So repeat different values of the independent variable, calculate means, etc. And then what's been measured. So like I said, sometimes people put an extra M into the QORMs. So there might be, because there could be, more than one thing that's been measured. So what's been measured before, what's been measured afterwards, what are the time intervals, how are you doing the measuring? So are you using a gas syringe, are you using a measuring cylinder, are you using a balance? What's been kept the same? So we got examples of temperature, light intensity. So rather than use something vague like keep the conditions the same, we need to be specific about what is being kept the same. So temperature, mass, light intensity, etc. So good practice is students often begin an answer to a planning question by writing out this acronym QORMS, and sometimes next to it they will put the relevant aspects, and then they will go ahead and write their answer. So this is a very, very good framework. It works well, and students generally are good at it. And invariably, even if they're a weak student, they can still pick up marks on a planning experiment just by writing something such as, repeat this, repeat that, etc. So something simple. They can always pick something out and pick up perhaps 1 or 2 marks. So using QORMs makes the planning experiments accessible to all. So we have this question here, another design question. This is looking at it's another glass house question, but using QORMs, students can pick up marks, even if the situation is very unfamiliar. So there's a reference here to temperature. So this is looking at, changing the temperature. Will it produce a significant increase in crop yield? So there's an indication here, well, I would need to measure the crop yield. So that could be measuring mass, for example. So QORMs can be applied to a very, very wide variety of situations. Okay, we have a question related to this, which is when students are describing an experiment, is it acceptable to describe according to the way they've done their school in their school? Or should they describe it exactly as explained in the Pearson textbook? So I would say that students should describe a method as they have done it. often, doing is the best practice. Students often remember how they've gone about and done something. So there is no problem in describing the method that the student has experienced themselves. They do not have to specifically learn the Pearson descriptions, the Pearson methods. So it's up to, well, perhaps you as a teacher giving guidance and perhaps the student's preference. As you're probably aware, there are videos available of these core practicals. So perhaps some students will watch a video and they're able to recall sort of steps of a method from that video. So to answer this question, it doesn't matter how the method is described, as long as it fits in with what we're looking for in terms of that CORMS framework, what's been changed, what organism has been used, repeating, what measurements been taken, etc. It doesn't matter. There are many, many different ways of describing an investigation or designing an investigation, all of which can score top marks. So there is no one fixed way of addressing this. It could be a whole variety of different methods that could score full marks. So it doesn't have to be what's in the Pearson textbook to get full marks. So here we've got the mark scheme for this particular question. So controlling or changing different concentrations of carbon dioxide. The EQ means equivalent, so they might say something equivalent to levels or amounts of carbon dioxide. Using same plants, same species, repeating their concentrations, measuring the mass, or how many seeds produce, number of leaves, and so on. So here we've got another mark for measurements. So measuring the mass after a stated time, perhaps, do it after a week, do it after a month, or wherever might be relevant. And then what's been kept the same temperature, the same amount of light, using the same type of soil, same sort of watering regime, given the same amount of water, same volume of water, using the same mass of fertilizer on the crops, etc. So the students can pick up marks using that CORMS framework for this investigation. They don't have to have experienced the investigation. It's a matter of applying what they know of planning experiments to an unfamiliar situation. And then we have another one, which is looking at the effect of vinegar on the digestion of starch. Include details. You'll notice that these questions all end in the same sort of command. Include experimental details in your answer and write in full sentences. They both say the same thing. So that's typical of one of these experimental design questions. So by practicing, students become familiar with what's expected, really good for perhaps a homework or pre-exam practice, etc. And again, you've got the mark scheme. So what you're changing, so the concentration of vinegar, etc. Perhaps measure the pH. Organism. So we've got reference to starch. So that's what's been used. It could be flour, because obviously flour contains starch, or bread or potato, etc. What's been repeated? So repeat for concentration of vinegar. Iodine solution to test for starch. Again, keeping the temperature the same. Same time for the reaction, using the same volume of amylase for the digestion, etc. So again, the CORMS framework is being used in the mark scheme. So you can share those mark schemes with students and show them how the answers have been marked. So the command words, we've already talked a bit about these. So command words tell students what to do, and obviously it's important to get students familiar with how they should answer questions. Sometimes there's confusion between describe and explain. There is, across all the international GCSE sciences, there is a common taxonomy, which is in the appendix of the specification. And as we've already said, the type of command word links to the demand of the question. So AO1 tends to be the lower demand command words, and AO2 and AO3 tend to be the higher demand command words. So here we've got, it might be a bit difficult perhaps for you to see, but we've got the, this is the taxonomy. So you can use this to teach students the main command words which are used in the exam questions. So describe, for example, give an account of something. Statements do not need to be developed. They're often linked. There's no reasoning involved. And if we look at explain, an explanation requires a justification or exemplification of a point. The answer must contain some element of reasoning. This could include mathematical explanations. So in a describe question, there is no reasoning. It's just a matter of stating facts. Whereas an explain question, there is some kind of reasoning has to be given. So that's a key difference. So it's useful to share this taxonomy. I don't think we should expect students to learn a definition for every single word, but the key ones that crop up in the question, such as describe, explain, evaluate. Some of them don't really need any explanation, such as, for example, draw or complete. So as we've indicated, there is a sort of a hierarchy, a taxonomy of the taxonomy. So state and describe are less demanding command words. Evaluate and discuss are more demanding. So the AO1 questions would be less demanding command words, state, describe, and the AO2 and AO3 command words would tend to be more demanding, such as evaluate and discuss. That doesn't mean, though, that an AO1 question may not use a more demanding command word either. So how might you introduce command words and ensure students are familiar with them? Any comments? I'll give you a minute if you want to put anything into chat. I guess the main way that you might introduce them would be through different exam questions. So as you're teaching a topic, you might use exam questions to check knowledge and understanding of the topic. And as you use those different types of past paper questions, there will be different command words, and you'll be able to introduce them through those. So I'll give a minute. So that's probably nearly up now. If you've got any questions about introducing command words, put them into chat. Otherwise, I will move on. So we've been talking about AO3 in particular and CORMS for the experimental design. So we'll have a look at core practicals. So the core practicals, the practical work is embedded throughout the specification, and the points in the specification, which are italicized, these are the core practicals. So it's about developing knowledge. It's about developing hands-on skills. As I said earlier, I know that sometimes it's difficult, even in UK schools, sometimes to get hold of particular chemical reagents, and sometimes time constraints are a problem when it comes to practical work. But the students are expected to have knowledge of and to have done the core practicals in the specification. Sometimes it could be perhaps through a teacher demonstration. But the core practicals are expected to have been covered in one way or another. I say there are often there are videos available of these as well. So the core practicals are assessed within the written papers, as we've already said, and practical skills make up to 15% of the total marks. So that's quite a proportion. And if the students actually do a practical, they tend to remember something about doing that practical, which they can then recall in an exam question. We got one response to the command words. Responses says that they do more essay questions to make students familiar with command words and make them understand them thoroughly. So thank you for that. So practical skills are very, very important. So here we've got, I know in the previous slide it said about italicized, but here we've got some practicals, core practicals, which are in bold and they have the B suffix, which means that these ones would only be assessed in paper 2. That doesn't mean that practical skills are not assessed in paper 1, but these particular ones that you see on the screen would only be assessed in paper 2 because they've got the B suffix and they're in bold. So you've got one about enzymes, one about energy content in a food sample, one about the light and gas exchange in leaves using hydrogen carbonate indicator, one about transpiration, and then the last one is about investigating the distribution of organisms. And as I said, certainly in some UK schools, that kind of practical work is difficult. The environmental type practical work is difficult to carry out, but there are always ways of getting around it, such as using small quadrats in a school classroom or laboratory. Some of these practicals may have been touched on even pre-GCSE as well, perhaps, for example, energy content in food. So in the international GCSE specification, there's no coursework. Neither is there coursework in the UK GCSEs. There's no practical endorsements, and that means that your head of centre does not have to sign a declaration that the core practicals have been covered by the students. As we know, the core practicals are assessed using exam questions, and the sort of techniques associated with those practicals can be assessed as well. And the apparatus and techniques list aligned with UK GCSEs. And I think we've already said that practical questions can appear, it can be covered by any of the assessment objectives. So the students have been asked to solve problems, apply their scientific knowledge to experiments, plan, which we've looked at in some detail, describe or demonstrate methods and safety procedures. So there was a comment earlier about students not being very good at safety. So that's something that just needs to perhaps be drummed into them, perhaps pre-GCSE and obviously during the GCSE course, because students need to know about how to handle equipment and materials safely. Variables, which is something, again, I'm sure that many of your students will be familiar with from pre-GCSE. Recording results methodically and precisely, analysing and interpreting data, using technical language, carrying out calculations and drawing graphs, and then thinking about accuracy, reliability, and validity, and evaluating methods and thinking about how could a method be improved. So that's a wide range of things that students might be asked to do. So a couple of questions to exemplify this. So a method has been described here. We won't go into it in detail, but students have been asked to think about, well, what is the dependent variable and a reason why the same volume of water is used. Then there's a calculation and a conclusion to evaluate. The conclusion is given, students asked to discuss it, and there are 4 marks for discussing that conclusion. So the discussion would have to include thinking about the results of the investigation and perhaps using the calculation from the previous part of the question, part C, the difference that's been calculated. So this is a situation where the method is given. Students have to think about, well, why have they done this? Why have they done that? So they're applying some of their knowledge from practicals that they've done themselves, identifying a variable, etc., etc. So that's a nice example of a question assessing practical skills. So we need to ensure that core practicals are completed during teaching. And ideally, the practical work needs to sit alongside and fit in with the theory work, but it could obviously be a standalone session. And students need to be familiar with recording methods, making their observations, writing down the results, and some kind of analysis. They can use a lab book if they want, class book, whatever suits you as a teacher. They don't need to use lab books. They can just, whatever normal methods of recording work is absolutely fine. So sometimes, depending on the practical, you might have some kind of carousel approach where you go to different stations and do something slightly different, or group work is fine. Students don't have to work on their own. Working in small groups is fine. It's all about gaining the experience of doing the practical work and being able to recall, what they did and why they did it. There are also suggested additional practicals in the specification, and but they don't have to be done, but they are there because it gives students more breadth to their practical experience, more unfamiliar contexts, but they don't have to be covered. And obviously, time constraints might mean that you do not have time to actually cover them. Just got a question come up about practical work. So is it necessary to carry out experiments for students in schools who cannot afford a laboratory? So if you don't have a dedicated laboratory, some practical work can be carried out in an ordinary classroom. But if you do not have the facilities at all, then using, well, whether it's a demonstration or whether it's using videos, animations, any way of exposing the students to practical work needs to be used, because one way or another, for this specification, the core practicals got to be covered. So ideally, we would like to see the practicals being carried out by students themselves. But if that really isn't possible, then any other method, whether it's a demonstration by the teacher, whether it's a video, an animation, anything to expose the students to the particular core practicals. So as I said, we've got a list of suggested practical work. As you can see, it's quite a long list, but to stress, these do not have to be carried out, but they are investigations that could be carried out to give extra exposure to practical work and to broaden knowledge, skills, and to give students experience of additional unfamiliar situations. I won't go through them all, but you see there's a long list, and these are in the specification in one of the appendices. So Appendix 6. So we need to think about embedding practicals in schemes of work. They can be used to reinforce scientific theory. And as I've already said, the students need to be thinking about, well, what am I measuring? Why am I doing it? How do I control variables? And some of these skills, this knowledge can be applied from one practical to another. What makes the results valid and reliable? And even if in the student's eyes, they talk about practicals not working. Any data can be used to learn from. So whether it's good or bad, if something didn't work, it can still be evaluated. So there's no such thing really as bad data. It's a matter of whatever data is collected, and it can be discussed, even if it's not perhaps the intended outcome. It provides opportunity for discussion and learning. There are some free guides available to download from the Pearson website. So we've got a guide on using practical terminology, and we've got a guide on the core practicals. So these are very worthwhile looking at. They'll give you as a teacher some guidance, and they're also very useful for the students to use as well. So we've got a pause for discussion if we need it, about preparing students for practical skill assessment. So we've already had one question, obviously clearly where there is an issue regarding carrying out practical work. But if you've got any points you'd like to make regarding preparing students for practical skills assessment, please let me know in chat. Otherwise, I will move on. I'll just give you a moment now if there's anything you want to say about how you prepare students for practical skills. Like I said, I do appreciate that in some schools, both in the UK and abroad, practical work can be difficult to manage for different reasons, whether it's the availability of a laboratory, whether it's the availability of materials, etc., etc. Okay, so let's move on. So if you've got anything you want to put into chat, please do. Otherwise, we'll move on. So we've already had one question pop up, but let's have a think now. Let's move on to math skills. So maths is an integral part of biology. The way that these skills are assessed is they're in some kind of context, not just standalone questions. And you can see there's a large proportion of marks which are linked to maths skills. So we've got math skill supporting areas such as the analysis of data, calculations from experiments, and drawing graphs, evaluating patterns. So confidence in maths will support the performance of students in AO2 and AO3. In the appendix of the specification, there's a full list of the skills, but these include percentages and ratios, averages and estimates, using standard form and significant figures. So for example, a question might say, calculate this to X number of significant figures, or write your answer in standard form. Plotting and interpreting graphs, typically they might be worth 4 marks or so for different things such as, scaling the axes, drawing the axes in an appropriate size, labeling the axes, plotting points accurately, drawing an appropriate line through the points, etc. And then we've got the final skill included is rearranging equations. So these need to be used in context during teaching, such as if you're teaching enzymes, you can have questions related to calculating the rate of a reaction, calculating the size of a population. If you're using microscopes, calculating a magnification. There are lots of situations that mathematics can be used and assessed throughout the biology specification. Obviously, some topics lend themselves better than others. Using maths earlier than GCSE is important. So Key Stage 3 science, different math skills can be introduced. And then when the students get to GCSE, what they've done at Key Stage 3 just becomes normal practice and not unfamiliar. You can use starter activities. You can use exit tickets, in other words, perhaps a slip of paper with a question or two on it at the end of a lesson. If you've got a practical-based lesson, you can have data handling tasks within that lesson. So just different ways of making students familiar with mathematics, using data, handling data, calculating things, different types of calculation, rearranging equations and things. Just short activities to routinely introduce maths into science, into biology, and to make students more familiar and more confident as they go through the specification. As we know, for some students, the mathematics is challenging, but the more we use it and the different range of activities that we use will help to make students more confident, hopefully. So again, I'm not going to through this list, but this is for biology, chemistry and physics. So the ticks show the specifications where the skills are required. So you see for biology, there's a large proportion, all their data handling skills are required in biology. Some of the more complex skills are not required in biology, such as 4E in the graph work. But nevertheless, there are a large number of math skills required in biology. So like I said, these are in the maths section of the biology specification. A couple of examples. So students are given a diagram of a leaf on the left here. There's an arrow drawn on the right of the diagram, X to Y. Students are told that the actual thickness is 1,100 micrometers. They're given a conversion of a millimeter to micrometer. Determine the magnification of the diagram. So they're expected to measure with a ruler in millimeters from X to Y, and then use the equation to calculate the magnification. Worth 3 marks, so there'll be a conversion mark from millimeters to micrometers, for example, obviously a mark for the correct answer. I always stress to students that in this kind of question, they should measure in millimeters and not in centimeters. Measuring in centimeters introduces extra layers of complication unless they're good with converting numbers. One thing to bear in mind as a teacher, if you print questions such as this, depending on the scaling of the printing, the answer that is calculated may be different to an answer on the mark scheme. So the answer on the mark scheme is based on the full size printed paper. But if the scaling of your printing is different, then the final answer will be different. Just so in other words, if you reduce the size of the diagram compared with the original, then the answer for the magnification is going to be different. Just something to bear in mind because it can cause confusion. And then the second question is about calculating a percentage increase. So they look at the graph, look at the differences. So this is a matter of teaching how to calculate percentage change. So 2 very different types of mathematics questions. And earlier I showed you that we have guides for scientific terminology and the core practicals. And we also have a mathematics guide as well, Maths for Scientists. So again, free to download from the Pearson website. Useful for you as a teacher, useful for students. Again, we have a discussion question about how you might best prepare students for maths in a scientific context. If you've got any thoughts about that, put them into chat. I'll give you a moment, give you a minute. Okay, so yeah, so comment about using um, maths questions to enhance the skills. So practice different styles of question. So ideally, questions linked to put them in context for what you're teaching. Um, and also we have a comment about asking for help from maths teachers. So sometimes you might need perhaps to get some advice from your maths teacher colleagues about the best way of teaching a particular maths skill. So that's always useful. Sometimes the maths departments teach skills in a different way to us scientists. So sometimes it's worth getting some comments from maths teachers to find out how they go about things. But practice, practice, practice in context is a very good way of ensuring that students become familiar. They're not phased by maths questions, and it just becomes routine, and they're expected to see them. Okay, so we've looked at the assessment objectives. We've spent some time talking about practical work, and we've just been talking about math skills. So we'll have a look at how we can plan the course. Okay, so we need to start with the scheme of work. Apologies, I've moved the slide on. You can obtain a detailed scheme of work from Pearson for International GCSE Biology. And the scheme of work is editable, so that you can use it to support different ways of delivery. So, for example, some centres might be delivering the GCSE in just 1 year. Typically, it will be obviously a 2-year course. We've alluded to the fact that the content can be delivered in a modular way or in a linear way. So with the modular, it's effectively smaller chunks of the content are assessed rather than the content being assessed all in one exam series. Students might, in year 1 of the course, do modules covering some content, and then in year 2 of the course, they would do additional modules to cover the whole of the content. And then the marks for the different modules are put together to come up with a grade. It's a slightly different model. It doesn't, if you remember earlier, we said that in the linear model, raw marks are used to arrive at the grade. That's not quite the case in modular, but I won't go into that now to overcomplicate things. But it's easy enough for you to get the modular specification and make comparisons. But the modular model is becoming more popular. shall we say. So the scheme of work can be customized to suit how your timetable works, and how you break down the curriculum, and obviously the needs of your students. So it's a very detailed document, very useful. And if you're just starting out, it's something that you can make very good use of. And then as you become more familiar with it, you can start to move things around in terms of, looking at your school terms, thinking about, times of year when you might do particular kinds of practical work. So, for example, in the UK, we would be unlikely to do the ecological type practical work in our winter time. So we would look at teaching so certainly aspects of topic 5 when the weather is warmer. So we can fiddle about with the content of the scheme of work and the time of year it's delivered. You can access this from the Pearson Qualifications website, which is fairly easy to navigate. So, depends obviously how big your school departments are. You might be a single teacher, you might be a bigger school with several budget teachers. And depending on the ability level of the students you teach, you could make changes to this scheme of work to suit the students and to suit your particular way of working. So that document is something that's free and should be very, very useful to you if you're just starting out in particular, but still useful if you're an experienced teacher. The textbooks are matched to the specification. As I said, the specification was released in 2017, first examination 2019, but there have been additions to it since then, different versions, and the changes to previous versions are shown in one of the first few pages of the specification. It tells you which version you're looking at, and the changes since the previous version. Usually there are small changes, perhaps to a specification point or some other aspect of the specification. There's something called Active Teach. So these are digital materials, paid for materials, which can be very useful. Wide variety of content. So different worksheets, different assessments, the sort of tests and things. So all of the active learn materials align with the specification and the assessment objectives. So if you've got the resources available to purchase, there are sample materials that you can look at for Active Learn before making the jump and making the purchase. But they're very closely aligned with the specification. And if you are just starting out, a very, very good source of materials to use in lessons. And again, some of them can be edited as well. So it's best to use the scheme of work as a foundation, and then you can add in your own practicals. For example, if there's some practical that you're very familiar with that you've done and sort of tried and tested, you could insert that instead of one that's there. You can add in your own assessments. So using the scheme of work as a sort of a backbone, as a foundation, you can add things to it. You can delete things that are there and add things in just to make it personalized. And that's the idea of it. It's not something that you have to sort of go through specifically, sort of step by step. You can edit it, you can modify it to suit your own situation in school, your own experiences, and just make it personalized. And then there is a course planner that shows the different options for delivery. So whether that's over 1 year, whether that's over 2 years, is it modular? And there's also a mapping document. Somebody's also already mentioned the Cambridge International course very early on in today's presentation. But the mapping document looks at the content that overlaps with AQA, which is another UK-based examination board, Cambridge International. So if you are moving from one exam board to another, the mapping document allows you to see the content that is familiar with perhaps a course that you've already been teaching, and will obviously show you the differences. So useful particularly if you're changing from one exam board to another. Coming back to the scheme of work, it highlights specification points and links them to suggested learning activities in lessons. And there are things for you as a teacher and for students as well. Plus, there are links, there are hyperlinks and things to resources, be they sort of printed resources or videos, animations, that kind of thing. So there are lots of resources available to you, in addition to obviously purchasing the textbook, which is quite a commitment if you're just starting out teaching a new course. But you can certainly familiarize yourself using the Pearson website. You can familiarize yourself with all the materials that are available. And like I said, for example, for the Active Learn, there are sample materials available for you to look at. I've certainly used them myself, and I can vouch for their quality. So this is just a summary of what kind of materials are available to you on the Pearson website. So you've got sort of guidance on assessment, course planner, exemplar material, mapping document that we just mentioned, the maths and practical support. So those practical guides, etc., that we saw earlier. Past training content, so you can access prior or previous training courses, guides on the qualifications, support with results, the scheme of work, etc., etc. So lots of materials available to guide you and help you. And at the very bottom of that list, it says transitional material, which we've just been referring to, sort of that mapping document and so on. So, and transition from sort of the Key Stage 3 into GCSE and GCSE onto A-level. So we do try to support you. So here we have just an example of a lesson from the scheme of work. And as we said, you can fully edit all of these. So we've got structures and functions in living organisms. This is topic 2. We're looking at gas exchange. We've got 3 specification points listed. 2.46 about the thorax, 2.47 about intercostal muscles, the diaphragm and ventilation, and then 2.48 about alveoli. So some suggested activities, making a model of the thorax, labeling a diagram of the alveoli with an adaptation, so that's quite useful. Large diagram of an alveolus with labels highlighting the adaptations of the alveolus, and thinking about how the adaptation increases gas exchange. Using demonstrations such as a bell jar model, or if you haven't got one, a physical model, you can use an animation of that. Lung dissection. Students often find that quite informative, but obviously it's not something that as a teacher you may be comfortable with. And then we've got in the 4th or 5th column the references to the student book. And we've got references to a teaching hub, which I'll mention in a moment. And we've got sort of worksheets and guidance for teachers and students on the bell jar model. And then the last two columns are about the kind of skills that students might develop from this lesson, problem solving, etc., creativity, reasoning, and so on and so on. So in terms of the long dissection, so you've got a skill about ethics, integrity, and so on. So that's a typical page that you can fully edit. So you can put in your own examples of practical work, your own links to worksheets, etc., etc. Your links perhaps to other books other than this Pearson book, whatever you have available. So typical page, fully editable, and so you can sort of work through, plan your course, use materials that you've already got, add in materials that perhaps new to you from the different Pearson resources. Okay, so we spent quite a lot of time talking about different types of question. So have a look now at mark schemes. So as I said, I'm an experienced biology examiner, principal examiner. So I've been heavily involved in writing papers over many years, writing mark schemes. So here we have a very straightforward example of a question with the mark scheme. So the command word in the question is state. And the question is about the incidence of mutations being increased. So in other words, what can increase the incidence of mutations? So radiation, UV light, tar, tobacco, a mutagen, a carcinogen, gamma rays, X-rays, radioactivity. All mark schemes have an additional guidance column. For some questions that might be blank, but often it's not. So here we've got additional guidance. Do not accept infrared radiation. And that's because infrared radiation does not cause mutation. We'll accept smoking, and we would accept other correct mutagens. So a student might name a mutagen that you're not familiar with, so you would have to check that out. But any other correct mutagen would be awarded the mark. Just a bit of background about mark schemes. So when we write an exam paper, we have to work to the framework of getting the percentages of AO1, AO2, and AO3 questions correct. We also write the mark scheme at the same time, but then sometimes students don't always answer questions in the way that perhaps we as examiners anticipated. So once an exam has been sat, the senior examiners, we look at quite a large number of responses from students, and we look at the mark scheme that we've got, and we then might make some changes to the mark scheme, because sometimes students come up with things that we hadn't thought about. Sometimes they don't come up with the things that we had thought about. So the mark scheme tends to evolve a little prior to marking. So like I said, we look at a large number of responses as a senior team of examiners, and we look at what we will and we will not accept. So we start with the framework of the original mark scheme that was written at the time the exam paper was written, and then we make tweaks to that mark scheme so that we are marking all student responses fairly. Because like I said, sometimes students come up with things that we hadn't thought about as a senior team of examiners. Sometimes we have points on the mark scheme that we simply don't see. So we might move those to perhaps the additional guidance under the accept heading. But mark schemes do change a little bit prior to the main marking process. Another example of a question in a mark scheme. So this is using a different command word, explain. So in an explain answer, we are looking for some kind of reasoning. So this question is about stem cells being used to treat blood disorders, such as sickle cell anemia. The question tells the students that sickle cell anemia affects red blood cells, and it tells them that leukemia affects white blood cells. So explain why stem cells can be used to treat a variety of blood disorders. I did say earlier that in an explain question, quite often a mark might be available for stating some correct fact in relation to the question. So if you look here, we've got about stem cells can differentiate. So that's a statement of fact about stem cells. And the second point on the mark scheme says what they can differentiate into. In other words, different blood cells, various blood cells, any type of blood cell. So you notice the word blood is underlined. So we're looking for the word blood if we're awarding a mark in the second point. So they are statements of fact. There's no reasoning going on there, no judgment being made. But later on, we've got the reasoning. So for example, the fourth bullet point, they can make white blood cells and destroy pathogens, or they can release antibodies, or they can make platelets and help with blood clotting. So there you've got the reasoning. They can do this and they can do that. So the reasoning is the reason why they're used. So stem cells can make red blood cells, and red blood cells transport oxygen. So if you've got sickle cell anemia, which affects red blood cells, using stem cells means that red blood cells can be made, and those red blood cells can transport oxygen. So a mark, possibly 2 marks, could be obtained for some kind of statement of fact, but then to get the full mark, full 3 marks in this case, there has to be some kind of reasoning. So the third, the fourth, and the fifth bullet points here include some kind of reasoning, and they do this, and they do that. So if there's no and, no mark would be awarded. So I think I've probably explained most of what's on this slide already. So as I said, the principal examiner who's writing the exam paper will write the points on the mark scheme, what we expect students to be saying. We don't expect the exact wording, but we are looking for key vocabulary to be used. We're looking for clear answers which convey information, biological information correctly. The additional guidance column tells us what we will and will not allow, gives some examples of some correct answers which are not in the main column, and might also give the examiner some instruction. So if it says ignore, you just treat a word or a phrase as if it's not there. So just imagine that you would cover up that writing and just ignore it. Sometimes we use do not allow or reject. So that would be if a student has used a word incorrectly, for example, in the incorrect context, in a sentence, then we would reject it. So, for example, if it's about cell division and they're writing, perhaps they should be writing about mitosis, but they write something about meiosis, and some other comment, then we would perhaps we would reject it. So those comments, those do not allow and reject, are not used that frequently, but in some questions they need to be used, because otherwise we would perhaps be awarding marks for incorrect biology. Underlined words. So we already saw some underlined words, the word blood in a previous mark scheme. So they must be included in the response. If a word is in bold, we need the word that's in bold or a synonym of that word. Smooth brackets. If I just go back to the mark scheme. So here, for example, the first point on the mark scheme, blood stem cells is in smooth brackets. So blood stem cells comes from the question. That's how the question starts. So on the mark scheme, we put that into smooth brackets so that the whole sentence sort of reads better. But the fact that those three words are in a smooth bracket means that the students do not have to write that in an answer. They could just say, they differentiate. That would be fine. But in the mark scheme, we put those extra words in so that so it reads better. But the students don't have to write those points. Curly brackets. Now we haven't seen any curly brackets, but sometimes we have curly brackets, various words within those curly brackets, and the answer must contain one of those words or phrases. So these hopefully are useful points for you as a teacher to interpret the use and application of a mark scheme. We have clear or key principles. If the scientific idea is clear, we award the mark. If the vocabulary is used accurately and the point is in alignment with the intention of the mark scheme, we award the mark. So we are giving credit for correct biology in the correct context. If there's a misspelling, we would ignore it unless there could be confusion with another word. So we had those examples earlier. There was photosynthesis, and then there was that combined sort of mitosis, meiosis word, which we would not accept. Oops. So in the international GCSE papers, I've already mentioned this, but the questions are points-based, not level-based. So we saw a couple of 6 mark, the practical-based questions earlier. The list of points, so the more points students gain, up to a maximum of 6, that's fine. So the points-based mark schemes have discrete valid points. And I said that on the UK GCSE science papers, there are a small number of questions which have what we call a levels-based mark scheme. You'll come across those at international A-level biology. Excuse me. So the mark scheme will tell you what's credit worthy for each question. So for a 6 mark question, there could be up to perhaps 10 or 12 points, of which 6 have to be gained for full marks. And the marks are for accurate scientific information and relevance. So it's not about how the answer flows. This is why we give the advice that using bullet points is good, because bullet points means that the answers tend to be concise, and then tend to be relevant. So they won't necessarily flow, but if there's a list of correct points listed as bullet points, then it's good technique for students to do that, because it means that it's easy for an examiner as well to pick out these correct points. So we do not need long essays to impress the examiner. We would rather, as an examiner, see bullet point lists which give clear, concise answers. So it's all about get your point across clearly, structure your answer well. Don't try to write long sentences which could end up in confusion. Okay, so we're very close to the end of the presentation now. So I'm hoping that you as teachers have well, obviously gained some information about the course, the structure of the International GCSE Biology course. And if you're just starting out or thinking of starting at some point in the near future, then it's given you some confidence to go ahead. You know that there's lots of information available to you from the Pearson website. So the scheme of work would obviously be one document that will be a very good starting point for delivering the course. But there is a vast amount of past paper materials available with the mark schemes. And also we haven't mentioned, but there are examiner reports as well. So as principal examiners, we have to write a report on the examination, and that includes examples of answers to questions with some comments about the mark awarded to that question. So the principal examiner report or examiner report is also a good source of information for you as a teacher to look at marked examples of questions and to learn about why they were or were not awarded a particular mark. So if you've got any points that you would like to ask, please put them into chat. I think we've probably already talked about the support that's available, but we've got obviously the Pearson website. We can start with, obviously you've got the specification that you can download. You can register your interest with Pearson. You've got all the course materials and the past papers and that kind of thing. All the support for you as teachers and the training support, so you can look at previous training sessions. There are published resources that you can access. You've got the guides for biology, for practical work, etc., and so on. And there are sort of news updates which are released roughly monthly, which can be accessed and you can read them. It gives you guidance about sort of new developments and timelines and that kind of thing. And there is an advisor who's called Tim Lawrence, and he has responsibility for psychology and international sciences. You can find his email address, and he can be contacted. So if you have any questions, he can be emailed, and his job is to advise you. But you can also sign up for those updates from the subject advisors. And there's also, as a customer, you can access a support portal. And there's also sort of community groups online as well. So there's lots of support available in terms of materials and contact with an advisor. Looking more deeply, we've got past papers available to you, which once you're registered, you can access these. Some of these obviously showing that the documents are locked, but as an advisor, you can if you register, you can access these documents, and you can see that this is an up-to-date document. We've got 2025 papers listed there with examiner reports, which I mentioned a few minutes ago. So lots of material available going back a number of years, going back to 2019. So lots of material because there are 2 sessions per year. Then you've got the teaching and learning materials and the training content. So accessing the Pearson website allows you to then access lots of material to guide you through the process of starting to deliver the course. Obviously, there's a lot to take in, but the help is there. So if you are not sure about something, my advice would be to contact Tim Lawrence, who's on this previous slide that I've just put back. You've got his email address. So if you email teachingscience@pearson.com and put Tim Lawrence's name in the subject line, and he will be able to answer that, any questions that you may have, and give you further support and guidance. There's also something called Results Plus, which allows you to analyze the performance of your students when they've sat an exam. So you get a breakdown of where marks were scored for individual students or groups of students. So you can pick out topics that were not answered well. You can pick out topics that were answered well. So picking out questions that, for example, were not answered well, you can then, going forward, you can adapt your teaching and perhaps the kind of support you give the students to improve teaching in particular topics. But Results Plus allows you to see the score for particular questions for an individual or a class. You can make comparisons with other classes in your school. You can compare against national averages. So Results Plus allows you to analyze performance and use that as a tool for progressing in the future. So the way Results Plus works is students take an exam. The exam papers get scanned. So they come to the UK and they're scanned. And then that allows them to be marked online. And then all the statistics of the marks for each individual part of a question can be analyzed, and that then allows you as a teacher to access the data about your students' performance. So you can see how they did on individual questions or parts of a question, and you can also see how that links with the different topics, so that then going forward you can think, okay, I perhaps need to teach this particular topic or part of a topic differently. I need to improve the way that I support the students so that they will perform better on these particular types of question. And then there's something called Exam Wizard that you may be familiar with. So it's another free tool. It allows you to, I know this says geography, but it allows you to access past papers. You can choose topics, so you could choose topic 1, 2, 3, 4, or 5. You can choose different assessment objectives, so AO1, AO2, or AO3. You can choose whether you want to look at maths questions or practical questions. So it's very powerful. And what you can do is put together a test paper, an exam paper, using different types of question style, etc. So it's very powerful. And you can put together perhaps a short test. You can just access a few questions. So if you want to do some maths practice, you can access some maths questions. If you want to do some AO3 practice, you can do some AO3 practice and perhaps look at assessment of practical design questions and that kind of thing. So Exam Wizard is very powerful and it works with Results Plus. So you can very easily use the back catalogue of exam papers to put together a very short assessment to check your students' learning, or perhaps a longer, perhaps mock exam type question covering a broader range of topics and assessment objectives. So it's very powerful. It's very easy to get carried away and put together something that's quite a large paper. So it needs to be used in moderation, but it's very useful, very easy to use. You can put together papers, you can store those papers, access them later, you can edit them. So if you're not familiar with it, it's very well worth looking at, very good resource. And then there's access to scripts. So access to scripts allows you to, once the marks have been, sorry, the grades have been released, it's an online service which you've paid for through the exam fees. You can look at mark scripts, so you can actually look at the mark script online. It allows you to, again, analyze what's good and what's bad about your students' performance, and you can use that information going forwards. And it also means that it allows you to think about whether or not as a centre, you're going to perhaps put in an appeal about students' marks. So if you want to have a mark reviewed, then looking at the paper first would give you a good insight as to whether that it's worth going ahead with that mark review. And Pearson's the only awarding body that provides access to these marked papers online. So you've paid the entry fees, the students, and you get this afterwards as a sort of resource. And then we've mentioned books, so paid for resource, student book, and gives you preparation for moving on to A level or an international baccalaureate or a BTEC. And you can access samples from the Pearson website. So as you were told at the very beginning, you should have already hopefully received a link, but there was a link put into chat. So you should have a copy of this presentation and things like this website for information and access to samples. You've got the link there so that you can access samples of the book if you want to have a closer look. And then we have another advisor, so Jonathan Wong, who's another advisor. So you can, again, same teaching address, Teaching Science. So just by contacting Teaching Science, you can get advice on what's new, what the new developments are, and so on and so forth about international GCSE biology and other international sciences. So, do we have any other questions? The Pearson host has just added the link to the slides on the chat. So the very last message on the chat has the link to the slides for today. So if you haven't received them, you can access them from that link, which hopefully, so you've then got the fallback if you want to look back and think about going forwards with this particular qualification. Okay, well, I'd like to thank you for listening and for contributions. We have a question about e-certificate. So I'm not sure whether Leon has any answers for that. So our Pearson host, you should get an e-certificate sent to you for attending this training. Okay, so thank you for your comments. Right, so in terms of so post-event, you'll be given your certificates, any issues. So there's a link there if you have any issues about so if you basically register for the event, you should, following this event, be sent your certificate for attending. But thank you very much. I wish you well, and hopefully it will be nice to think that you'll be going ahead and start to teach this qualification. So goodbye and have a good rest of your day.